a change in the second speaker. So Vicente couldn't make it, and instead it will be Barbara coming here. Well, I'm very pleased to start with Filipe França from the University of Bristol. Thank you, Filipe.
Thank you, Paloma. Good morning, everyone. For me, it's a privilege to be here to talk about our work in the Brazilian Amazon. So I separated my presentation into three main sections. First, a very brief introduction about Amazonia's importance and main threats. Then I'll follow that up by bringing some results from our long-term ecological monitoring program, using results from dung beetles to understand forest health. And then I'm going to end up bringing some examples of how long-term collaborations have helped us to embed evidence into decision-making. So why the Amazon is so important? With over 50% of our remaining forests, the Amazon is one of the really, let's say, containers of high density of biodiversity on Earth, including around 10% of vertebrate and plant species. But the importance of the Amazon goes beyond biodiversity. They also contribute directly for over 30 million people that directly depend on those forests for their livelihoods. The Amazon is also extremely important in a global worldwide dimension by helping us to mitigate climate change via net carbon sequestration, for example. So what are the main threats to the Amazon region? Deforestation, definitely, and it's on the rise for the last decade, particularly in the Brazilian Amazon. Remaining forests are also threatened by other human activities, such as logging, fires, the overexploitation of natural resources. And on top of those, the Amazon is also facing climatic stressors that act both at global and regional scales, such as warming temperatures, extreme events of floods, and droughts that are increasing in frequency and intensity at the region. So when someone is unwell, what do? We look for symptoms to try to propose health solutions. The same approach works for forests. Some of the researchers here today, they use, for example, tree mortality to understand forest dynamics, to understand how forest health is changing across time or across different scales. Other people are interested in understanding forest condition, for example, change in canopy openness, change in microclimate, or even in the temperature of the leaves in the canopy. Some researchers that like insects, like me, they work with insects. They use other biological indicators to understand forest health. In my case, our group used dung beetles and also the ecological process that those organisms do in the forests that contribute for the functioning of the forests. So before diving into our results of our research, I would like to give you a brief understanding of our experimental design. So in 2010, we went to the Amazon and we established around over 100 forest plots. Those plots were distributed within catchments, which are shown here in the orange. Each of those catchments received about between 8 and 12 forest plots. And those forest plots, they were distributed across a gradient of human activities, of human disturbance, including undisturbed forests, logged primary forests, burned primary forests, logged and burned primary forests, and also secondary forests, which are those that are regenerating in previously deforested areas. And at each of those plots, we collected dung beetles and the ecological process. From those pitfall traps, we were able to estimate change in species richness, species composition, abundance, and also in functional metrics, such as functional diversity, functional richness. And from those mesocosm arenas, we were able to estimate change in the process that the beetles do, such as seed dispersal, soil bioturbation, and dung removal, which is a proxy of nutrient cycling. So with that data from 2010, we were able to address two key research questions. First, how does forest disturbance affect dung beetle species richness, species rarity, and functional diversity? So to give you a brief summary of those graphs, in here you can see species richness across our forest classes, and here you can see species rarity, So the lower the number, the more dominated by rare species is a community. The higher the number, the more common are the species on that community. And on the right, you can see changes in in functional richness, which represents the functional space occupied by that community. So if you look on our primary forests, highlighted here in green, you can see that we found little effects of primary forest disturbance on dung beetle response. So that means that they were relatively resilient when it comes to disturbance within primary forests. However, the lower species richness, the higher amount of common species, and the lower functional richness in our secondary forests shows that deforestation resulted in major impacts on dung beetle communities and functional trait diversity. Our second research question using those data sets was trying to understand how the extinction of rare and common species would influence community functional structure. So how did we address that? Using data from undisturbed forests, we ran scenarios of species losses. We did that at two scales. First, at the local scale using plot level data, and second, at the regional scale using the total pool of species from those undisturbed forests. And we ran three scenarios. In the first scenario, we removed from the most rare to the most common species. Our second scenario, we removed from the most common species to the less common ones. And finally, we also run a scenario using species loss randomly. This is our new model. And we use this new model as the baseline to assess if those changes were different from changes by chance. And as you can see here, just to give you an idea of how can we interpret this graph, a loss of about 30% of the most rare species resulted in a loss of over 50% of the functional richness. On the other hand, when most common species are lost, the values were all higher than what we would find by chance. When we scale this up by assessing changes at the regional level, results were a bit different. So in gray here, we have the confidence interval. And as you can see, change or removal of the most rare or the most common species were within this interval, meaning that there was not a big difference across these three scenarios. So as to answering our question, yes, the loss of rare and common species can affect the community functional structure. However, those results those effects are scale dependent, and it's very likely that the regional functional redundancy may offset the impacts of species loss at the local scale. But then with these results, we are not really able to understand how things are changing across time, or how, for example, climatic stressors could be affecting biodiversity and ecosystem functioning in our study region. So to understand this third scientific question related to how previous forest disturbance influence the responses of biodiversity and associated functions to enzo-mediated drought and wildfires, we went back to our study region. And we were in a unique position because this region was greatly affected by the El Niño 2015-16. Adriani showed a photo of a forest that was affected by that. And as you can see here in gray, those are the areas that burned primary forests. So over 8,000 primary forests were burned in that region. And what did we do? We decided to go back to some of those plots that we established in 2010 and start monitoring how beetles would change across time. And in doing that, we selected 10 forest plots within each of those forest classes and having a balanced design between forests that were affected only by drought and both by drought and fires. And then we collected them again, both in 2016, 17, and now recently in 2019. So the results that I'm bringing here today is based on the comparisons between forest classes, years, and the El Niño classes. So if you look at here, you can see that across time, we found declines in species richness for most of the forest classes. The same pattern was observed for seed dispersal by dung beetles. So one of the key messages that we found is that both drought and fires resulted in the loss of biodiversity and ecosystem functioning from the dung beetle perspective, A second key message here is that we observed stronger declines in fire-affected forests, as well as that previous forest disturbance influenced post-El Niño beetle responses. So just to give you an example, in some forests affected only by drought, we observed a small recovery of species richness, while in those affected by fires, we still had declines up to 2 years after the fires happened. On the seed dispersal perspective, we observed, for example, way lower rates of seed dispersal in forests that were affected by fires twice, a while ago and during the El Niño, and when compared to forests that were affected only by drought. So before going to the next part, this study, although only for dung beetles, was used by local stakeholders to inform the management plan of one of the reserves where we do our research, which was one of the things that drove me, that motivated me to do the final part of my talk, which is related to embedding evidence into decision-making. So I would like to bring here the importance of building long-term collaborations with local stakeholders, not only to build healthy global scientific partnerships, but also to ensure that what we are doing here is translated to the ground, is translated to the real world where things are happening. And just to give you some examples of how this worked, we had the opportunity to participate in the working group that guided the management plan of the Tapajós National Forest. And by doing that, we were able to translate our research, our science, into practice, informing, for example, the areas where they would be logged, because this is a sustainable reserve. There is a community-led logging happening there, and also in terms of how to avoid fires to happening there. And this means that we need to engage with different stakeholders, from park managers, from ICMBio, which is the Biodiversity Conservation Chico Mendes Institute, responsible for managing all the reserves in Brazil. We also need to get in contact with local communities and with the FUNAI, and also indigenous leaderships. So engaging and translating science means that we need to contact and engage with many people at different places and different types of actors. Another thing that results from those interactions and long-term collaborations includes, for example, co-organizing stakeholder events. For this one, for example, we had the Brazilian Forest Institute, representatives from different NGOs, local university researchers and students. This also gives us the opportunity to co-supervise students and publish with them, as well as recently we produced this book about the biodiversity from those 2 forest reserves. So the key points here, based on those examples, on those experiences we had in the Amazon, is that embedding evidence into decision-making needs a global thinking. We need to address the global issues we are having, but we need to also take into consideration the importance of local actions and local collaborations. Embedding evidence into decision-making also needs long-term commitment and continual, clear, evolving communication with diverse actors, with different stakeholders. And this is really important to ensure a mutual understanding of the needs and also of the expectations of those involved in those partnerships. And finally, embedding evidence into decision-making needs equitable partnerships with diverse authors to ensure that different voices are considered, as well as that the decision-making power and the leadership are shared. Thank you.
Thank you, Filipe, for this amazing talk. It was very nice. I'm